Hi everyone and welcome. My name is Gesine Heuck and it is my pleasure to host and present today's webinar entitled Nano-sized Delivery Systems for Vaccines. For a short background on myself, I am a pharmacist by training and I earned my PhD in pharmaceutical sciences at the University of Geneva in Switzerland. I've accumulated 10 years of experience in the development of drug delivery systems and translational research by now, with a strong focus on nanoparticle formulation with various systems and types of materials. In my current role as manager for customer service at Precision Nanosystems, I am in charge of the service and support programs to users of the nanoassembler technology. Before we begin with the actual webinar, let me mention some basic housekeeping points related to the GoToWebinar control menu that you will see on your screen. If you have technical issues, please use the question and chat box and we'll do our best to troubleshoot. You can also use this chat box to ask questions or give feedback related to this webinar. I will try to address as many questions as possible at the end of this session, but if I do not get to your question, we'll make sure to follow up with you via email. So make sure to provide us with a valid email address. With this, let's get started. The aim of today's webinar is to give you a high-level introduction to some of the recent advances in the vaccine field that got me, or I should probably say us, at Precision Nanosystems quite excited. And that's not only because they feature a strong relation to nanomedicines, but even more so because they show how fast-moving and dynamic the development of vaccines has become. Here we'll address the relationship between vaccines and nanomedicines, and then we'll illustrate this with some concrete examples of vaccine adjuvants, then vaccines aimed at preventing infectious diseases, and then we have the therapeutic vaccines that are mostly used in oncology. And then, of course, this will all be followed by concluding remarks. I should mention that this webinar will not go in depth into the molecular biology of vaccines. However, if you do have questions in this regard, please let us know and we'll do our best to answer those questions. Now, if we look into the history of vaccines, which is, by the way, over 200 years old, we can see that research has become or has come a long way to improve the safety and efficacy of these vaccine agents. We started off with live vaccines, which consist of an inactivated or attenuated infectious organism. Then we moved to vaccine subunits, or rather subunit vaccines, that feature only part of the pathogen, for example, proteins, that will then serve as antigens. And these vaccines, or these subunit vaccines, have a lower safety risk than the live vaccines. In the next generation, we get the recombinant vaccines that typically involve inserting DNA that encodes for an antigen into a host cell, and this host cell will then generate the antigenic protein. The use of DNA bypasses the need to produce and purify proteins in vitro, and with that, it accelerates the development process, and of course, it also reduces the cost significantly. So that was a good step forward. And finally, in recent years, the use of mRNA to generate vaccines has become a hot topic. And in contrast to DNA, mRNA cannot insert into chromosomes of the host cell. And so that additionally reduces the safety concern around the occurrence of mutations and oncogenesis. Now, going back to the historical context, I think it's worth to note that with time, we have also learned not to only improve the type of vaccines, but also we've learned about how to use vaccine approaches and how to take advantage of our immune system to modulate it for our immune response, not only for preventing diseases, which is what we do with preventive vaccines. So here we administer a vaccine or vaccine agent before a disease occurs. But now we also have learned how to exploit our immune system and modulate our immune system with therapeutic vaccines. And these vaccines are administered when a disease is already occurring or existing in our body. And this is, for example, the case for cancer and HIV. One thing I haven't mentioned yet is that while these newer vaccines here have improved the safety profile with respect to these live and inactivated attenuated vaccines, but they're also associated with lower immunogenicity and lower stability. For example, a naked protein or nucleic acid are both susceptible to degradation by proteases or nucleases, respectively. And so you have to find ways to appropriately deliver them to the host cell. And also, because they have lower immunogenicity than the traditional live vaccines, you may need to, or what's often required is to add additional components that we call adjuvants to actually stimulate this immune response. And what comes in very handy here, or what can be very beneficial, is to actually use nanoparticles or nanomedicines to combine these advantages or the requirements that would be protection of a susceptible antigen and stimulation of the immune system in one small delivery vehicle. And similar to small molecule delivery, for example, you can exploit these nanoparticle delivery systems to combine really all these advantages. For example, if your antigen or your nucleic acid is sensitive, you can encapsulate it on the inside of this nanoparticle and make sure it's only released to the host cell at the right time and in the right amount when it gets there. You can also attach the antigen or an adjuvant on the outside of this nanoparticle to expose it to the host cell or to expose it to immune cells to trigger an immune response. You can take advantage of the material that you use in this nanoparticle, and you can use this material as the actual adjuvant. This is, for example, the case in the first example I will name, where we use, or where the researchers use, the researcher group uses a lipid-like adjuvant that they incorporate into a liposome bilayer. There are also polymers that you can use as a matrix for nanoparticles, but at the same time they function as adjuvants to trigger an immune response. And last, you can also use these nanoparticles and modify the surface in a completely different way. For example, you want to have a stealth-like system that is not immediately recognized by the immune system, or you need to stabilize your particles, so you can attach stabilizers such as a PEG lipid or a PEG group on the outside to give this entire vaccine particle here a longer shelf life, for example. And moving to our first example that I already mentioned shortly. This is a project or a work that was done at the University of Strathclyde by the group of Yvonne Perry. And Yvonne's group has focused a lot on the exploration of using or preparing nanoparticles to combine adjuvants together with an antigen in a way that triggers a safe immune response and an efficient immune response in the body. And one of the components that Yvonne's group focused on was the trehalose dibehanate, in short TDB, which is an immunostimulator. So TDB is actually a lipid-like material, But TDB cannot be formulated into a liposome by itself because it immediately aggregates. It's pretty fussy. And so the idea was to combine this TDB with another lipid, which is a cationic lipid in this case, to then also allow the adherence or the attachment of an antigen. And in this case, they used the model antigen ovalbumin, which is slightly negatively charged, so it could interact with the DDA. And so they combined all these lipids, and in some cases, they used an additional stabilizer lipid, here cholesterol. They combined all of these in the microfluidic system to prepare these liposomes, and then they added the antigen to the surface of this liposome to do some tests. And in our next slide here, we see first on the graph on the left-hand side, we see the different formulations that Yvonne's group prepared. So they had different combinations where they had first only the DDA, so the cationic DDA and the TDB. Then they added different amounts of cholesterol in there to see how they can prepare particles, or rather liposomes of different size. And they varied the size by varying the parameters, the flow ratio parameters in the microfluidic instrument, the nanosimilar instrument. And then they compared what kind of different sizes they could prepare. And they also compared this to a control particle that only consisted of a neutral lipid together with a DDP. They also looked at the surface charge here, and as you can see, the liposomes containing the cationic lipid, of course, had a more positive surface charge, and then the liposomes containing only the neutral lipid had a surface charge or a zeta potential close to 0. Then took some of the lead candidates out of this lot and loaded it with the protein. And you can see here that the loading efficiency of the liposomes that all contained the cationic DDA was very good, which makes sense because the negatively charged protein interacts with the cationically charged lipid, whereas the control liposome with the neutral lipid had a very low loading efficiency of the protein. From there, they took a lead candidate and injected it in vivo in mice. So they injected it intramuscularly with the goal to look at the recruitment of monocytes in this mouse model. And the way this works is that the monocytes are stained in blue. And so when there's recruitment to the injection site, you should see a blue stain in this muscle that you can actually see here in this little picture. And then they also looked at the retention of their antigen or of their component at the injection site. And this was the first control injection here. As you can see, they only injected the antigen. And you can see that the retention over time was very low and it decreased very quickly. You can also see that the recruitment, there may have been a bit of recruitment of monocytes, but it wasn't very significant. They then injected the antigen in combination with the DDA, and you can see a significant increase of the retention of this antigen at the injection site, and the same as the DDA, the cationic lipid. You can also see here from the blue stain that there was significant recruitment of the immune cells. And this recruitment was enhanced even more when they combined the DDA and the antigen with the immunostimulator TDP. Now, there are, as I mentioned before, many other types of vaccine adjuvants that you can use. And I wanted to mention just a very preliminary result here that was generated by my colleague Sham Garg, which just shows the feasibility of encapsulating antigens into polymeric nanoparticles, which are often used in the vaccine development. And Sham, what he did here is he prepared PLGA nanoparticles and played around a bit with instrument parameters of the microfluidic system. And he was able to prepare particles of different sizes just by modifying the total flow rate in the microfluidic system. And he also got a very nice acceptable polydispersity. And he also, after loading, obtained a nice encapsulation efficiency of 20%, with total loading of the protein of 5%, which is what is the standard in the literature. And this research is ongoing. In my second example, I wanted to touch on the development of nanomedicine vaccines for infectious diseases. And this work here was done by a collaboration between the group at MIT, Dan Anderson's group at MIT, and a daughter company of Moderna, which is called Valera. And Valera focuses solely on the development of vaccines, mRNA vaccines against infectious diseases. You know, in this project, the two groups prepared mRNA containing lipid nanoparticles, where they combined hemagglutinin mRNA with lipids into this lipid nanoparticle, and they used it as vaccine. And they combined the mRNA, the hemagglutinin mRNA of the pandemic strain, either of the H10N8 pandemic strain or of the H7N9 pandemic strain of the influenza virus. And for those who are not familiar with the influenza virus so much, hemagglutinin is a component of the virus capsid of the influenza virus that the virus actually needs to attach to the host cell. Now, after formulation of these particles of this vaccine, the group injected these vaccines into rodents. And this study was done in mice here. And here we see the example for the injection of the H7N9 Influenza Virus vaccine. And they injected this mRNA lipid nanoparticle vaccine at different concentrations, as we can see here from the different colors. And then they challenged the mice after 7, then after 21, and also after 84 days, and looked at the effect of the immunization. And what we can see here from these survival curves is that all of the vaccines or the vaccinations that were done with this mRNA vaccine outperformed the control groups in the survival. And even after 7 days, we can see almost complete vaccination, even with the lowest dose of administration. And this study was very successful also in primates. And it was successful enough that the group performed a pilot study in humans. And in this case here, we see the example of the vaccine against the H10N8 influenza virus, where they injected this vaccine into humans. And then they looked at the hemagglutinin inhibition titer and also the microneutralization assay, the titer. And as you can see here from these results, the vaccinated groups showed significantly higher titers than the placebo group. In another collaboration with a number of other academic institutions, Valera also worked on, or is still working on, developing an mRNA vaccine against the Zika virus. And here they actually did a very similar thing as they did before. They combined the mRNA into a lipid nanoparticle. And this time the mRNA that they used encoded for the PRME gene, which is also, it encodes also for a protein that is part of the membrane of the Zika virus, in fact. And one thing to know about Zika vaccines is that there is a high concern that it triggers so-called antibody-dependent enhancement of dengue infections. And what that means is that if a person is vaccinated with the Zika vaccine, this may actually trigger a higher response to a dengue virus infection, which could be lethal. And so this group was very clever. They went ahead and just modified this wild type PRME mRNA to prevent this cross-reactivity from happening. And they tested both of these lipid nanoparticle vaccines in rodents, and they got a very good response in both cases, where they were able to prevent the Zika virus disease. And in the case of the modified RNA, they were also able to prevent the dengue virus antibody-dependent enhancement. And in the case of the wild type mRNA, they in fact achieved the Zika virus sterilizing immunity. The take-home message for me from both of these studies was that it is very exciting actually how fast the groups were able to develop these vaccines. And this is specifically important when you think about preventing infectious diseases. Especially if you look at the pandemic influenza viruses, they can spread within weeks or several months. It took this group a couple of weeks to develop this vaccine, which stands in stark contrast to the traditional methods of developing vaccines, which can take months. So from the moment that the pandemic strain is identified, if you can develop that vaccine within a week or two, this really prevents this pandemic from spreading, which I think is a significant progress in the fight against some of these infectious diseases. My last example touches on the use also of RNA vaccines, but this time we will focus on the treatment with an RNA vaccine. So here we talk about a therapeutic vaccine that is being applied for the therapy against glioma. And this work was done by a group at Arcturus, a biotech company that is based in San Diego, California. And this group has their own proprietary lipid nanoparticle delivery platform. And they formulated specific mRNA, a microRNA called miR124, into these lipid nanoparticles. And they then, or the idea of this vaccine is to apply it and to stimulate the immune system, so stimulate white blood cells and modulate them. In this case, we're talking about PBMC cells. And these cells will then go into the brain and target existing glioma cells. This group developed the particles and also injected them into mice. So they tested them in vivo in mice. And what we see here in this graph, it's also a survival curve. As you can see, the formulation with the LUNAR nanoparticles outperformed all of the other control formulations, which consisted of different types of lipid nanoparticles. And one of them was also a formulation where they combined the microRNA with lipofectamine. What we can see is that it outperformed the all these control particles and achieved better survival of the mice over time. What I do find very interesting in this study is that on one hand, it really points out the potential of using microRNAs as therapeutics, which is a fairly new field, but a very interesting field. But I also found in this study that it was an extremely elegant way to target the brain, which is really normally a very complicated organ to target. On one hand, due to the blood-brain barrier, but also on the other hand, due to just other occurring side effects when you try to actually reach this tissue. With this, I'm already at the conclusions. I do hope I was able to convey some of these messages that I listed here. The first one being that nanomedicines have become an integral part of the vaccine development process. And I'm sure we will get a lot of new exciting publications in the coming months and in the coming year. The second point being that the onset of novel RNA lipid nanoparticle systems allows prompt responsiveness to pandemic threats, which is really a great step forward in improving global health. And the third point being that nanomedicines open new opportunities for therapeutic vaccines, hopefully in the future also going beyond cancer and HIV. With that, I would like to thank you all for your attention and for listening. We are open for questions now, so please don't hesitate to send them our way using the chat box. Thank you.